ranked by this THEQS university ranking. Have you heard of it? And what do you think of it? Should we worry too much about what the THEQS World University Ranking say about our universities all over the world? Thank you.
Okay. This also, I would say, is a complicated question, and the answer is probably yes and no. The external rankings, be they of universities or of scientists or of individuals in any walk of life, are complicated because they do provide an element of external validation, while at the same time many such organizations, and I don't know enough about this one to make any specific comments, many such organizations tend to be rather conservative. So they look towards really past achievements in the context of what has been done and what has been known, rather than looking and really evaluating future potentials. So, for example, in judging science, some of the most innovative science has been rejected by some of the most visible scientific journals. Why? Because innovative science is not popular, because nobody is paying attention to it. And it's only some years later, after it's been established, that the community recognizes that what was done was fundamentally important and truly pioneering. I think the same possibility exists with organizations of this sort. They will obtain criteria, but the criteria may not be the most forward-looking. On the other hand, there is the external validation that can be very important in drawing faculty, in drawing students, and in recognizing accomplishments. So I think one wants to basically be very conscious of these issues and not compromise internal standards and internal goals, but at the same time be sufficiently pragmatic to not ignore external agencies of this sort. And again, I say that with limited knowledge of this particular group, but just as a general sense of external validation as a route to quote, success.
Thank you very much, Professor.
Thank you. Yes, please. There are two. Sarah, can we give to the ladies first, please? Yeah.
Okay. Well, good morning. I am Dr. Gisela Gonzalez from the Center of Molecular Immunology in Havana, Cuba. First of all, I want to thank very much to Dr. Horvitz for being one of these persons that have spent his own life or his life in working for the benefit of the people. And then I want to ask Professor Horvitz some opinion about our work. In this case, I am going to refer to our personal work in our center, our Institute of Research in Havana. I will want to hear his opinion in some part of this work. We have spent a lot of years in working for new therapies targeting cancer. I mean, we have, in this case, we have approach of targeting the epidermal growth factor receptor system. That means we are trying to target the cell proliferation quality of cancer cells. In this case, as you have referred, cancer is one of these diseases where the apoptosis do not play its role, and then this comes together with uncontrolled growth. In our case, we have been working for several years in the development of a vaccine that targets the epidermal growth factor, that is the ligand. And then this vaccine provokes an anti-EGF antibody response, and this antibody blocks the binding between the EGF and the receptors, and then in some way stop the proliferation of these cells. This has been our work for several years. By now we have concluded phase 2 and phase 3 trial, And we have demonstrated that there is an increase in survival for patients that are being vaccinated when compared with the randomized control that do not receive vaccination. In fact, already now we have registered this vaccine in our country and is available for all advanced lung cancer patients. My question is the following. What we always expected from this vaccine or from this kind of therapy is to have like a stop in growth, and because of this, like a prolonged survival. In fact, we have demonstrated that. We have, I'm sorry, we have prolonged survivals in patients that receive the vaccine, but not when compared with patients that does not receive the vaccine. But we have also observed several cases. I can mention around 10 patients in a population of 700 patients that we have already with this vaccine. About 10 patients that have not only a stop proliferation, but have a tumor regression. That means in some moment their apoptosis begin to work. And how we always have to try some explanation for this, because we are not only stopping proliferation, we are provoking cell death with this kind of vaccine. And then we always thought that in some way there should be like a kind of equilibrium that among apoptosis and cell growth, and that in cancer what happens is that the cell growth is more than apoptosis, and because of this, well, the tumor grows. And in this case, as we stop cell growth, the mechanisms go towards the apoptosis again. But well, this is just an hypothesis. We have never been able to test that. And I would like to know your consideration about this. What do you think about this? Why we are getting not only prolonged survival, but also disease, tumor regression, tumor remission, disappearance of tumor. That means that apoptosis is working there again. I would like to hear about your comments about this. Okay. Thank you.
Okay, I'm not sure I heard all of the specifics there, because there was some resonance with the microphone. But I gather the question, at least in part, is with this antibody that's been developed, you find that you can both block cell death and stimulate apoptotic programmed cell death. And in this way, you're actually intervening with the two sides of cancer, because as I described, cancer on the one hand involves too much cell division, and on the other hand, too little cell death. So if you can block division and amplify death, you're taking advantage of both worlds. Now, there are certainly data in the field, not from my laboratory, that indicate that there are particular human cancer genes, like the gene MYC, that is involved in both the proliferation and in the cell death. And so if one alters basically the control appropriately in cells, you can change from what would be a driving towards cell division to a inhibition of cell death. So I think what is known about the biology of cancer would fit very well with what you've just described, but I don't know if this particular gene would necessarily be the one that's important. The idea of using antibodies to intervene in this way is, I think, one of the most exciting areas of modern biomedicine, because it used to be, not very many years ago, that all of the drugs that were dealt with were very small molecules that were identified by pharmaceutical companies that were very much chemistry companies. And what has evolved over the past 20, 25 years with the emergence of genetic engineering and biotechnology is that there are now drugs that are so-called biologics, not little molecules, but much bigger molecules, and molecules that actually can be made using DNA as the basis of the synthesis of the molecules. For example, proteins such as antibodies or growth factors or other proteins. And the advent of biologics in clinical medicine has offered a whole new world towards therapeutics. So I find the area very important and very exciting. And of course, if one thinks about what's going on commercially, also very timely, as you may have noticed, that the leading, or at least one of the leading biotechnology companies, Genentech, has just been acquired by one of the major pharmaceutical companies, Roche, because Roche has recognized and has been interested for some years that this whole area of biologics is much the future of human drugs.
Thank you very much, Professor.
Thank you. Okay, we may have two more questions. We give to Professor Tengku Sara and Professor Lulaiming. Please.
It was a great honor to listen to your very clear and also very inspiring talk, Professor Horvitz. I am from the medical faculty, orthopedic surgery actually, so we are very, very applied really. And I would like to ask you, firstly, how or should we be getting into basic research, or should we leave it to those who are very inspired, etc., rather than the medical doctors in applied research? Actually, it's very attractive because I wouldn't agree that it's nothing, because impact factor, etc., actually favors basic science quite strongly. So we would like to get into this, but maybe you can give us some advice. And secondly, we are also probably in this country a little bit too fond of jumping on the bandwagon. So for instance, nanotechnology, stem cells, etc., when it comes in, we try to get involved, but sometimes a little bit too late. And maybe you can advise us how to start afresh and create something which is new on our own. Thank you.
Thank you.
Okay, so the first question is a question that I think can be asked at many different levels. Individually for a researcher, departmentally for a department, institutionally for an academic or hospital institution, and nationally. I don't think it is by any means necessary for every individual to do basic research. In fact, I think that would be quite counterproductive. And there are only a few individuals who have both the expertise and the talent to do both the basic research and the applied research. So I'm thinking more generally at the level of departments, institutions, and countries to have the interface, but one wants there to be communication so that those people who are doing the more basic research are interfacing with those people who have the more applied expertise, so that they're not acting essentially in vacuums, but rather are talking, interacting in a synergistic